Yeah. In today's class, we are discussing the 2023 previous year question paper of PG entrance botany. So the question is the expanded form of ELISA is what is ELISA? The full form, that is enzyme linked immunosorbent assay. You know, for detecting the antigens, ELISA is an immunological detection test. Okay, antibodies are linked with the enzymes. Okay, so phosphatase, such enzymes are used. It produces some color reaction. So based on the color, they estimate the this one. So quantity of the antigens. Next, about 50 to 15 to 20 vascular bundles are found in the outer rings of the anomalous secondary growth of. So anomalous secondary growth we see in Boerhaavia, Echinaanthus, such things we see. There is a deviation. Okay, there is a deviation from the normal secondary growth that is called as anomalous secondary growth. Here we see more rings of vascular bundles. In case of the Boerhaavia, we see the normal, apart from the normal outer ring of the vascular bundle, we see two more rings of vascular bundles. So that is called as anomalous secondary growth, where we see 15 to 20 vascular bundles. Okay, so that is the right answer. In Boerhaavia, we observe more number of vascular bundles, 15 to 20 vascular bundles in the um, outer rings of this. Okay, so that is the answer. Next, ilachi, haldi, idicium belong to the family. So cardamom, it is also called as Electeria cardamom, and curcumalanga, haldi. The botanical name is curcumalanga. So hidicium. So these belong to the family Gingiberaceae. Gingiberaceae is also called as family of spices. Many spices belong to the family Gingiberaceae. So the right answer is Gingiberaceae. Next, the plants growing on saline soils are called as, xerophytes, which grow in the desert conditions. Okay, next, halophytes. Halophytes is the plants which grow in the saline soil. That is the right answer. Mesophytes are the normal plants which grow in the normal conditions, and hydrophytes are the plants which grow in the water. So the right answer is halophytes, which grow in the saline soil. Next, which of the following activities prevent afforestation in the deserts? That means, which prevent afforestation means growing the plants in a higher scale, large scale. So goat and grazing animals, local residents, low rainfall, infertility of soil. So in the deserts, the main problem is with the low rainfall. So that is the nearest answer to this. Due to the low rainfall, there is not much growth in the deserts. Okay, next. What is not true for rhodophyceae? Rhodophyceae, we know in the algae there are different classes: chlorophyceae, phaeophyceae, rhodophyceae, like that. So what is not true? So we have to check for the ones which are true, and out of the four, which is not true. Absence of centriole and flagella. So it is true, because there is no flagella. In case of the gametes or the spores, we don't find the flagella. In case of the rhodophyceae, for example, Polysiphonia is the best example for the rhodophyceae, where we don't find flagella, and also centrioles are not there. So that is true. But they are asking for the wrong statement. Okay. So next one is accessory pigments include phycocyanin, phycoerythrin, and allophycocyanin. So that is also true statement, because phycocyanin, phycoerythrin and allophycocyanins. These pigments are present in the rhodophyceae. Okay, due to the presence of these pigments, they appear in red color, particularly. You see red sea. Why that sea is called as red sea? Because due to the growth of the red algae, it is dominant. So that sea appears in red color. So here pigments are present, phycocyanin, phycoerythrin and allophycocyanin. So that is also right statement. They reproduce using alternation of generation. That is also true, because we already discussed alternation of generation means there are two stages in the life cycle, gametophytic stage and sporophytic stage. So that is also true. And the stored food is Floridian starch, very similar to cellulose. It is true that the storage form is Floridian starch in the rhodophyceae, but it is not true that it is similar to cellulose. Cellulose is the material present in the cell walls. So they should have given. It is very like a very tricky question, because we may think that it is true, but it is wrong, because it is similar to starch, not cellulose. So this is the wrong statement given out of the four statements. So we have to choose the wrong statement. What is not true for rhodophyceae? The wrong statement is this fourth one. Next, male and female gametophytes are independent and free living in. So they are asking for the alternation of generation. We see two stages. One is sporophyte and another is gametophyte. We already know that male and female gametophytes are independent. They are asking that male and female gametophytes. So here mustard, we see both mustard is angiosperm and cast also angiosperm, and they have the bisexual flowers. Both the stamens and gynoecium are present in the same flower, and castor also, they have bisexual flowers, and pinus, male and female cone are present in the same tree or plant. So in the sphagnum is the right answer for this. It is a mass. Sphagnum is a mass. That means it's a bryophyte, where male gametophyte and female gametophyte are separate. Even in case of the Markamsha, they may give Markamsha. In Markamsha also, that is also right, because we see both the male gametophyte with the anthridiophore and female gametophyte with the archegoniphore. They are separate. They grow independently. Sphagnum also like that. So right answer is sphagnum. Next, which of the following ecological pyramids is generally inverted? So we see the ecological pyramid. There are three types of the ecological pyramids. Okay, pyramids of mass, pyramids of biomass, pyramids of numbers, and pyramids of energy. So these are the three different types of ecological pyramids. And here you should remember that they ask some tricky questions on this, particularly which one of the following pyramid is always upright? They ask like always upright means it is straight, it is not inverted. In case of the energy pyramids are pyramids of energy. In pyramids of energy, the pyramids are always upright, straight. But in case of the pyramids of mass, biomass, and pyramids of numbers, in some cases, in few cases, we see inverted pyramids. Okay, so where we see this one, I will show you in the diagram here. You see here in the pyramids of biomass, they are asking about the pyramids of biomass in the aquatic system, in the marine ecosystem, in the normal ecosystem, in the grassland ecosystem, or in the normal ecosystems, we see this is the normal upright pyramid. Okay, pyramids here, pyramids of biomass. Here you see producers, primary consumers, secondary consumers, and tertiary consumers. So here the biomass is more. The biomass of the producers is more compared to the biomass of the primary consumers, which are herbivores like grasshopper. And biomass of secondary consumers is less compared to primary consumers. And finally, biomass of the tertiary consumers is even very less. So we see the upright pyramid in case of the grassland ecosystem. But in case of the marine ecosystem, right, in such cases we see the producers, though their number is more, but their biomass is very less. It is the least. Phytoplankton, their biomass is the least. And next, herbivores, their biomass is more than the producers. And primary consumers' biomass is more than the herbivores. And secondary consumers' biomass is even more than the primary consumers. So we see the inverted pyramid in case of the marine ecosystem. In the marine ecosystem, the pyramid of biomass is inverted. And also one more thing they didn't ask, but in case of the pyramid of numbers, also usually we see the upright pyramid, straight pyramid. But in case of in some cases like the parasitic ecosystem, in the parasitic ecosystem, the pyramid of numbers is inverted. So these two instances you should remember. For pyramid of biomass, marine ecosystem is inverted, and for pyramid of numbers, the parasitic ecosystem. In case of the parasitic ecosystem, the pyramid of numbers is inverted. So these two examples you have to remember. Okay. Now the question is about which of the following ecological pyramids is generally inverted. That is pyramid of biomass in a sea or marine ecosystem. They may ask sometimes marine ecosystem or sea. In the sea, the pyramid of biomass is inverted. That is the right answer. So pyramid of energy, it is always upright. There are no examples for the inverted pyramid in case of the pyramid of energy. Okay. Pyramid of numbers in grassland is also upright. Pyramid of biomass in a forest also upright. So the only inverted one is pyramid of biomass in a sea or marine ecosystem. So which among the following is the correct order of classification? Correct order of classification they are asking. Like in the classification, we see the first kingdom, next division, next class, next order, family, genus, and species. So this is the right order. It is also called hierarchy of classification. Okay. Next, which is known as fossil bryophyte. So we know Markamsha, it is a living bryophyte. Pixie also living, and Markamsha polymorph also living. The only fossil form is Nyadita lanceolata. So this is the fossil bryophyte. Fossil bryophyte is Nyadita lanceolata, that you have to remember. Next, in a DNA molecule, they are asking, they are giving some statements. We have to choose the right one. In the DNA molecule, there are two strands, right, which run anti-parallel, one in 5' to 3' direction and other in 3' to 5'. So that is exactly anti-parallel means. Anti-parallel means one strand in a direction 5' to 3' direction, other strand, opposite strand in a reverse direction, that means 3' to 5'. So that is called as anti-parallel. Now, so if both are 5' to 3', it is called as parallel. But one is 5' to 3', another strand is like this, another strand in reverse order, like 3' to 5'. That is called as anti-parallel double helical structure. So this is the right statement. Okay, that we can understand. Next, Sachs recognized three types of tissue systems in the year. So in which year Sachs identified three different tissue systems? We know three different tissue systems. One is epidermal tissue system, ground tissue system, and vascular tissue system. These are the three types of the tissue systems, and which were recognized by Sachs in the year 1875. So that is the thing you have to remember, 1875. Next, the following ratio is generally constant for a given species. So in the this is also related to DNA, where the ratio of the nitrogen bases they are asking. So here A plus T and G plus C. So this is constant. So as per the Chargaff law or Chargaff rule, the number of pyrimidines equal to number of purines in any DNA. The DNA, since this is double stranded, always A form the hydrogen bonds with the T, and G always form the hydrogen bonds with C. That means A is a purine and T is a pyrimidine, and G is a purine, C, that means cytosine is pyrimidine. So as per the Chargaff rule, the number of purines is equal to number of pyrimidines. That means A plus T equal to G plus C, the ratio. So this is true statement. Next, Carper-Kappe theory was proposed in 1917 by who proposed Carper-Kappe? So this is related to root apex organization. This theory explains the root apex organization. So who proposed it? Schupp. Schupp proposed the Carper-Kappe theory, and Schmidt, what he proposed? He proposed Chuenka-Karpus theory. Okay, that is related to shoot apex. That is also related to shoot apex, but it is a different theory, Chuenka-Karpus theory. And Hofmeister, what he proposed, already we know apical cell theory. It was proposed by Nageli and supported by Hofmeister. We can call both Nageli and Hofmeister, they are related to apical cell theory. And Bouvet is French theory. Okay, so French concept. So you have to remember Carper-Kappe theory was proposed by Schupp. It is related to root apex. And apical cell theory also related to root apex and shoot apex. Next, Cranch anatomy was firstly observed in 1914 by 1914. Who observed for the first time Cranch anatomy? It is related to C4 plants. Okay, in C4 plants, we observe in the leaves, we observe a particular type of anatomy that is called as Cranch anatomy. So that was observed by Haberland. The right answer is Haberland. Cranch anatomy in C4 plants was observed for the first time by Haberland. That is true. Next, in which of the following plants strobili are not found? Strobili means cones. Where we see the cones? In Equisetum, we see the cones. In Cycas, we see the cones, male and female. And in the Pinus also, we see male and female cone. But in case of the Diapteris, cone structures are not formed. We only see the sporophylls. That means sporangia are attached to the leaves. In case of the Pteris, in case of the Diapteris, we see that the sporangia which form the spores attached to the leaves, these are called as sporophylls. So cones are not observed in case of the Diapteris. That is the right answer. Next, the anther wall layer, which is usually polyploidy and has dense cytoplasm as well as high DNA content is. So with reference to anther, TS of anther, in TS of anther we observe several layers, epidermis, and we see the stomium, middle layers, tapetum. So all these endothecium, all these are the structures from the epidermis, endothecium, middle layers, tapetum, and microsporum. We observe these structures in case of the anther, and here you see, yeah, in the structure of the anatomy of the anther, the cross section, we see it is a dithecous condition here. Here in each lobe there are two microsporangium. Here there is the outer wall is called as epidermis. Next is endothecium. Next middle layers. Next tapetum. So here and inside we see the microsporangium with the microspore mother cells. Finally, they form the after meiosis. They form the microspores that is called as microsporogenesis. Okay, diploid microspore mother cells undergo meiosis to form the haploid. Each mother cell forms 4 haploid. Each microspore mother cell after meiosis forms 4 haploid microspores. They are also called as pollen grains. They are also called as pollen tetra. Okay. So here there is a layer surrounding the microsporangium. It provides the nourishment to the growing microspore mothers. It provides this layer that is called as tapetum. It provides the nourishment, that means nutrients to the growing microspores or microspore mother cells. Okay, so there is a specific character about the tapetum. Usually in other cells we find a single nucleus, but here in the tapetum usually we find 2 nuclei. But sometimes polyploidy condition are also observed, and the dense protoplasm is also observed in case of the tapetum. So the right answer here is what they ask, the anther wall layer, which is usually polyploidy. The only layer is with a diploid condition or polyploidy condition is tapetum only, and has dense cytoplasm as well as high DNA content is tapetum. That is the right answer, tapetum. Next, heterochromatin code was introduced by. So with respect to DNA, we see euchromatin, which contains the active DNA, active genes. But with respect to heterochromatin, we find the inactive DNA. Okay, when it is stained, it appears in a dark color. Heterochromatin appears in the dark color, whereas euchromatin appears in the light color. Okay, and who introduced the word heterochromatin is the right answer. Is the right answer. He first introduced the word term heterochromatin. Next, in a stamen, proximal end of filament is attached to. So, with respect to flower structure, we see here. This is the flower. This is stamen. This is called as proximal site or proximal end of the stamen. This is the distal end. Proximal end is attached to the petals and sepals, and distal end is attached to anther. So the right answer here is proximal end of filament is attached to thalamus or petals. So that is the right answer, proximal one. Next, 4-nucleate monosporic type of embryo sac was first discovered in. So we know monosporic type. There are 2 examples are given, polygonum type and Enothera type. So here polygonum contains, but polygonum, it is monosporic type of embryo sac, but it has the 8 nuclei type. 8 nuclei are present in polygonum. But next one is Enothera type, where we find only 4-nucleate condition. Both are, polygonum is monosporic type, Enothera also monosporic type, but Enothera contains 4-nucleate condition. 4 nuclei are present in embryo sac of the. Next one, Enothera is the right answer. Next, large empty colorless cells present on the addoxal epidermis along the veins of grass leaf soil. So we see the cells with very thin cell walls and large in size, which are present in the epidermis of the grass leaves. Okay, so those are bulliform cells. Okay, these are also called as motor cells. They are useful in curling of the leaves. When there is lot of heat, there is a lot of sunlight, then the leaves are like a string, they curl. So it prevents the excess transpiration, it prevents the loss of water. So these cells, bulliform cells, are present in the grass leaves. They protect the leaves from the excessive transpiration. Next, in plant tissue culture studies, polyethylene glycol, it is also called as PEG, is used to induce. So we know that in presence of the polyethylene glycol, there is a fusion of the protoplast take place. Protoplast fusion. So that is the right answer. Protoplast fusion is the right answer. PEG or polyethylene glycol, it induces the fusion of the protoplast. Next, in higher plants, the chloroplasts are. So in which shape? We already discussed spirally coiled, spiral structures in Spirogyra, reticulate form in case of the Eidogonium. Okay, but in case of the higher plants, angiosperms, we find the chloroplast in a disc-like form or discoidal shape. That is the right answer. Next, linker DNA binds. We know already we discussed in the nucleosomes. If there are two successive nucleosomes next to each other, the DNA which connects these two nucleosomes is called as linker DNA. Okay, so what it does, it connects two successive nucleosomes. It connects two successive. That means the nucleosomes which are next to each other are connected by the linker DNA. So that is the right answer. Next, which one is wrongly matched? Here you see uniflagellate gametes. They are asking for the wrong state. Okay, uniflagellate gametes in polysiphonia. It is a wrong statement because polysiphonia, where we don't find any flagell, either in case of the gametes, okay, or in case of the spores, we don't tetrasporophyte. For example, from the tetrasporophyte, tetraspores are produced, and here we don't find the flagella in case of the gametes. So that is the option we should give. And next, in a cell, tRNA constitutes about. So they are asking about the percentage ratio of. There are three types of the RNA: tRNA, mRNA, rRNA. Where rRNA occupies most of the 70 to 80% or more is rRNA, ribosomal RNA. It is also called as rRNA. So it is around, it is more than 80% in the cell, and 15%, 10 to 15% is tRNA, and 1 to 5% is mRNA. So the right answer is 15% of the total RNA is occupied by tRNA. Out of the total RNA, tRNA occupies around 15%. That is the right answer. Next, cell plate which appears during the cytokinesis ultimately transforms into. So we know during the cell division, karyokinesis, cytokinesis. The karyokinesis means division of the nucleus. And next we find the cytoplasmic division, cytokinesis, where initially we observe the cell plate, formation of the cell plate with the help of the Golgi complex. Okay, and later it is transformed into middle lamellum due to the incorporation of the calcium and magnesium pectate. It forms middle lamellum. Okay, that is the right answer. Next, barochory is a phenomenon of. So it is related to dispersal of the seeds. The dispersal of the fruits which are fallen due to the gravity, high gravity, is called as barochory. The fruits with the high weight, they are fallen down and seeds are dispersed. So that mechanism is called as barochory. The mechanism of seed dispersal. There are different kinds. Hydrochory, that means dispersal of the seeds with the help of the water. Hydrophily is different from hydrochory. Both involves water. Okay, that is a pollination. Hydrophily means pollination with the help of the water. Hydrochory means dispersal of the seeds with the help of water. Okay, so anemochory, dispersal of the seeds with the help of the wind. And we also see in some cases there is a explosive mechanism. Egbolium, in case of the egbolium which belongs to Cucurbitaceae, where we see the fruit explodes so that seeds will be just like a bomb. Fruit explodes and all the seeds are dispersed with very high speed. That is called as explosive mechanism. Example is ecbolium. Okay, so you have to remember all these. Next, in plants, leaf fall is due to the production of the. It is related to phytohormones. Abscisic acid. It is a right option because it like it is a negative growth hormone. It induces the leaf fall and it induces the dormancy, seed dormancy or bud dormancy, and also it protects the plants from the high transpiration. Okay, when there is scarcity of water, immediately abscisic acid it enters into the leaves and it closes the stomata. It is useful in the closing of the stomata to prevent the excessive transpiration because both are required, just like switch on or off. So this abscisic acid acts like a off condition. Okay, so leaf fall is induced by abscisic acid. Next, Crohn gall disease is caused by agrobacterium. We know agrobacterium tumefaciens. It causes Crohn gall disease, particularly in the dicots. And next, in a dihybrid cross between yellow round seeds and green wrinkled seeds, uh, the phenotypic ratio in F2 generation. Okay, it is a dihybrid ratio. We know the phenotypic ratio is always 9:3:3:1, and the genotypic ratio is 1:2:1:2:4:1:2:1. So they asked about the phenotypic ratio in F2 generation, that is 9:3:3:1. That is the right option. Next, the cells associated with the xylem and phloem at the end of veins are modified into. So they are transfer cells. They are parenchyma cells which are present at the ends, at the ends of the vascular bundles, particularly xylem and phloem, and they facilitate the entry of the ions in a large scale. These transfer cells are present at the ends of the veins. Okay, next. Duplicate of holotype is known as, we already discussed about the typification in taxonomy. We discussed about the different types. The type holotype is the original specimen which is explained by the author. But what is the duplicate of the holotype? That is the isotype. We already discussed about that. Isotype is the duplicate of a holotype. If holotype and isotype are not available, then we use lectotype. Okay, lectotype. It is the next one. It is a like a original, duplicate, triplicate. Like finally, if all these are not available, we use the paratype. So next, mannitol is the reserve food material. So we already discussed about different types of the reserve food material in algae. In green algae, chlorophyceae, the food is reserved in the form of the starch. And in case of the rhodophyceae, the food is stored in the form of the Floridian starch. But in case of the Phaeophyceae, the food is reserved or stored in the form of the mannitol or laminarin. Laminarin and mannitol, these are the two forms of the storage food in case of the Phaeophyceae, and fucus is an example. Ectocarpus, fucus, sargassum, laminaria, all these are the algae which belong to Phaeophyceae. So the right answer is fucus. Next, diacetic type of stomata are found in the members of. Where we find the diacetic type of stomata? There are, we already discussed. Okay, anemocytic, anisocytic, parasitic, diacetic, all these are the different types of the stomata. And in Malvaceae, we find the anemocytic type of the stomata. In the Brassicaceae, we find the anisocytic type of stomata. In Rubiaceae, we find the parasitic type of the stomata. And in Caryophyllaceae, we find the diacetic type of stomata. The right answer is Caryophyllaceae. They are also called as caryophyllaceous type diacetic. Next, one of the following organs penetrates into integumentary tissues and absorbs the food material. So this is related to embryology, where there are different types of the endosperm. Okay, where we find the this for the during the development of the embryos, there is a structure called as hastoria. Okay, it absorbs the food material. For example, so this hastorium absorbs the food material from the endosperm. Okay, and it provides to the growing embryo. That structure is called as hastory. Okay, that is the right answer. Next, mitochondrial matrix process. So in the mitochondria, we already know that in the mitochondrial matrix, the reactions related to Krebs cycle take place. So usually the enzymes of Krebs cycle are present in the mitochondrial matrix. That is the right answer. Okay. Enzymes of glycolysis, these are present in the cytoplasm or cytosol of the cell, not in the mitochondria. And enzymes of Calvin cycle, which are present in the chloroplast, not they are not related to mitochondria, because Calvin cycle is part of photosynthesis. And enzymes of nitrogen fixation also, they are not related to mitochondria. So the right answer is enzymes of Krebs cycle. They are present in the mitochondrial matrix. Next, sickle cell anemia is due to. So this is the like in the mutations, in the chromosomal mutations, you see chromosomal aberration, chromosomal abnormality, point mutation, frame shift mutation. Okay, point mutation is the right answer, because due to point mutation means the mutation at a single site of the nitrogen bases. Due to this, due to the mutation,